All right. How is everyone's progress going with the Fred Park table? Eh, okay. Yeah, so but you've established like a team and stuff? No. I think that's a big first step, right?
Yeah.
Are you, do you have an idea of who to talk to, or do you want help facilitating like
I
my
just need
team?
to look through the
Uh,
idea.
Okay, okay.
Honestly, just been super.
Yeah. Yeah.
So
Yeah.
planning on sitting out today and getting.
Yeah. Well, as again, like you don't have to just be loyal to one Fred Park table. You can like contribute to one, and then like just like jot your name if you contributed like at least a few ideas, not just like one and done. Yeah. Does anyone online have questions about the Fred Park table? One thing that we could do is I can make a tab on Notion. Um, let's see. Like on the ideas board, there can be a Fred Park per um like idea there, and then you can like if you contributed to it, then you can like put it on your name. For example, I'll just share my screen as an example. Uh oh shoot. I see. Okay. That is on my part. I've been a little behind on uploading the lecture video, so I realized that if people couldn't tune in live, then it'll be catching up. It's okay. Fred Park tables won't take too long, but let me do that. Let me put it high on my priority to upload those things. So um let me upload some, let's see. So the idea of Red Park comes from this guy named Alex Slocum from MIT. And it's just a systematic way that we are going to design our frugal science projects as a primer for those of you who haven't seen the recording I have not uploaded yet. But I'll upload this in the resources tab. But there's all these projects and ideas that are kind of like sparse in a way, or like they're descriptive of a bit high level, but we need to make it kind of tangible by making a table. So for example, I'm gonna go to Frugal carbon sponge for ocean sequestration in the ecology tab. Maybe I'll make a tab, or I'll open this all the way, and then I'll make a table. I think if I do slash table, I can make a table. So then I'll do like F R. I can add, what are the letters again? FRDP. Okay. Design parameter, functional requirements. Um, oh wait, can you get the door for me? Thank you. Um, analysis. Okay, analysis. Risks. No, references. Am I forgetting something? Okay, and then countermeasures. And countermeasures. And then you can copy Manu is almost on his way. Yeah. And then you can start populating ideas under each thing. So that's kind of what I'm doing. This one was created by Debioti Roy. So um yeah, just a way to, and then just so that I can track of who has done what, maybe you can put your name if you contributed to a Fred Park, you could put like contributed by, and then you could put a link to your page. For example, like I'll put Kevin. Yeah. So then now I know it's, and you could do like bi-directional link. So like you can just do something like contributed to, what was that one called? Frugal sponge, yeah, for carbon sequestration. So it's just a bi-directional link of how to do that. Cool. Any questions from online community? All right. Let's see. Maybe maybe for the folks here, we can start working on a bread park of your choice. Yeah, maybe that's a good use of time. Since does anyone want to? I guess we kind of did the choral stuff in person.
Debbie yesterday.
Uh-huh.
when we did-
Okay, awesome. That's awesome. Awesome. Jack, do you want to work on your Fred Park together real quick? Maybe we could do that together.
Sure.
Okay, is yours in the idea board already?
ours
Yeah, yeah.
Discord under
Oh yes, plant pulse. Okay.
yeah.
Oh, I just said.
Oh, get in the mix. We're still trying to kind of define a bit of the particular parameters.
Yeah.
Because the broad scope is to like render otherwise invisible aspects of my physiology visible. Main application. But we haven't really settled on particular parameters. Some of them end up being more expensive. Like, it would be cool if we could get an active reading of carbon sequestration. So generally, the essential amount of carbon dioxide. Good.
Do folks are there any Discord channels that you want me to create? Because I only realized that there's 4 projects, but there should be a lot more projects than this.
Probably set up the flow so that it kind of reverses direction. That. But the issue is if you want something educational, you don't want to.
Yeah, I got it.
Look at. So we're thinking of some other things. Like, Kevin, you know what the like cheapest, if you just wanted a narrow band wavelength, so 690,
Mhm.
what's like the cheapest filter you can
Do you need a filter, or can you just have or
It is for a complete.
okay, yeah, like a filter? Like, can't you just have like a thin film of like laminate sheet?
Would that work?
Or it would filter in a certain way, but not like the advanced like Door Labs filters. Oh, sorry.
Yeah, it certainly doesn't. Oh, you guys are the early risers. Oh, it's hard. Sorry about this. Is anybody
Yeah,
online?
there's a handful.
Few people
Yeah.
online.
Yeah.
Hey, everyone. Just move into the other room. It's like too small a crowd, but we can talk about projects.
Good morning.
Can you read what that says?
No.
I'll bring the sign in for a second. It's cute. Is instrument sign up sheet, blah, 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 through the end of the quarter spring, Mars Potato Growth Project.
Oh my goodness.
You know what, the movie?
Oh, yeah.
Yeah, Martian,
Yeah.
yeah. Okay, let's do a quick discussion on Fred parks and kind of other projects that you guys are thinking about. And then we'll cover, I just wanna record a lecture for other people. You guys started doing projects?
I think
You wanna, let's go around just briefly. We're going to start with a quick project discussion before diving into one area today. Who wants to go first?
I can start, I guess.
Yeah.
So, we started a channel on passive ocean monitoring,
Okay,
and there's been a few people interested.
Yeah.
I should check again the Discord,
yeah, yeah,
but I started at Fred Park, and I think people are contributing into it.
yeah. Do you wanna open it just briefly? To
Yeah,
show
sure.
everybody just, I think I'll tell you a little bit about what we did in the last couple of weeks in that space. And I went to Monterey day before yesterday for something. We picked a date, right, for Monterey?
It is May 30th work
Let's see, might be 2 days.
or May 23rd.
Let's take, maybe the ocean related people. You wanna just join Zoom?
Yeah.
That way everybody can see it. Oh. And then while you're joining Zoom, is there a specific question that's rising? Like in terms of thinking about, what kind of data being the highest
Yeah.
priority?
I can share my screen, Enrico.
Yeah,
So yeah.
Yeah, just,
So I noticed, so we're going back to the Argo floats
Yeah.
and the surface drifters. I noticed that they cost a lot. Argo floats, $20,000.
so Argo Flow, just the baseline construction, the plastic is 20,000. The actual construction of a functional Argo Float is. Sure. And it's a 4-year lifetime, and then it sinks to the bottom of the ocean. Somebody could get very rich, they should figure out how to bring.
Wait, so there's water at the bottom of it.
Yeah. It's better than littering floating in the surface. That's what people think. Because you could make them buoyant in its last dive.
If it was buoyant, couldn't you get it back?
The cost of finding and getting one is higher than deploying a new one. So I think it's, again, it goes back to cost. But passive is important. And when we're out at sea and you see Vela is just everywhere, passively doing their thing and disintegrating. Okay, so I like that. I like the direction. I think what's valuable in projects, we're just having a brief project discussion, is quickly narrowing down, although people will be excited about the nuance and the breadth of the area.
Yeah.
Slightly, gotta just kind of get a sense of like, oh, let's try if this is possible. And then that narrowness kind of brings a little bit of a structure that otherwise is hard, because otherwise everybody is interested in everything and possibility, but nobody is interested in one thing that can you say explicitly. A good Fred Park is you should be able to say, I have an hour, what can I do? Like immediately. And then you say, I have a day, what can I do? Oh, I have a week, what can I do?
And one avenue that I kind of want to explore is my friend, he's doing his lab is in the physics department.
Yeah.
They're developing a gas sensor, like a cheap gas sensor, using a single wavelength.
Yeah, is that a
And
methane
like an absorption, yes, like
it's
just like
Leo's
methane.
lab.
Yeah.
Yeah,
exactly.
Leo and I talk all the time.
Okay,
It would be phenomenal. It's a very clever idea. They're trying to do it for methane. And methane measurements also need to be made on top of the ocean, actually, too. And it's a free air sensor. So go ahead.
yeah, so my friend is building this out. I wanted to go visit it and see it.
Yeah, you should see it. Yeah, it's fun. I mean, I think I spoke to Leo, and the big conversation we had was about platforms. And the idea also works for CO2, by the way.
Okay.
And CO2 being measured right on the surface of the ocean is important, because now, surprise, surprise, there are certain portions of the oceans that emit CO2. Opposite Arabian Sea, for example, and ocean has 40x more carbon stored overall. So if it burps, it's going to be a big problem.
that's what we were talking about, because he was making measurements at Jasper Ridge on
Right.
methane, and
Yeah,
I was
yeah, yeah.
telling him it would actually be fun. So, surface measurements, even if they're atmospheric, but has to be done across, is kind of an interesting space.
Yeah.
That sensor narrows it down, because then you say, this is the specs of the sensor, let's build the platform.
Okay,
Let's just think about what does the platform look like. Starlink has changed the game for ocean sensors. I haven't seen that play out as yet. I'm saying it, sorry, I should say it should change the game, because it was so expensive to get data out.
Mhm.
And now we are out at sea and we're doing, I was teaching you guys. I mean, that is insane to think about.
Was that because of Starlink?
It's only because of Starlink. I hate the fact who owns it, but independent of that, in the old days, we used to be given a quota. You have like 5 MB of quota for your email per day. And if you cross it, it's shut down for everybody, like as a discipline. So now we're in the world of, so it's interesting. And that's another, it kind of, you see a crack in the puzzle and you say, oh, I'm gonna, so the specific question can be, is I have this type of a sensor, I have this platform in mind, and now we have this crack that Starlink is a viable strategy. So it could literally be a floating platform for anybody to build their projects on that gives you the Starlink bandwidth, something like that.
yeah,
And then suddenly you see like, oh, what I do is helpful for others, is helpful for me. And at that same time, I am narrowing down, I am using the word platform. So you let go of which sensor will be on.
okay,
If you're inspired by Vela Vela, then has to be a surface sensor.
Yeah.
So it's kind of, you can tweak in that, oh, we want an interfacial sensor. We don't want that. Suddenly you say, oh, I need a clever approach to make sure it is going to disperse. I want it to travel the currents. I want to be able to leave it in here and predictively. So then sail drones come to mind. There's a lot of these types of platforms that have been built. And then you also look at those platforms as your, so don't look at Argo then, because Argo is for depth sensing.
yeah,
So narrow it down. Passive, I'm just playing the discussion we had in the past. So I'm kind of narrowing it to surface,
Yeah.
surface only. I'm saying if it's surface, boom, you have communication for free,
right,
which had never been possible. I haven't seen an open source Starlink ocean platform. Sail drone and a couple things are like that, are there, and you should check what they do. And they have also become super expensive in some sense. People always have a tendency to keep increasing their platform, and the idea of you could shrink to see whether a project that had a total budget of say $5,000 or something. Like, I don't know what a Starlink antenna is. Maybe the subscription is in that range. And then you can start thinking a little bit about, oh, wait a second, I shouldn't just be saying Starlink. I should be saying, what is the technology underlying it? Maybe that's just, widely. And so it used to be that these satellite chips used to be very expensive. Okay, so that's a kind of a discussion I want to have with everybody. Anybody else who wants to go other than Danny, because we covered some of that.
Sure.
Yeah.
I'm with a couple people.
It's
Really,
fun to also have team names, by the way. So,
I believe we named ourselves Plant Pulse.
Flagpoles, I
We're on the
love it,
Discord. Check
love
us
it.
out. We're here.
Okay, who all?
So currently me, Megan, and Noe, I think are a couple of us who had
Got it.
chatted.
Yeah.
But we're still kind of ideating some of the particular broad goal is to visualize some components of plant physiology that are otherwise invisible. Getting into this idea that like the atmosphere, this idea of carbon sequestration is very abstract. The fact that we've gone from 200 to 400 ppm in carbon dioxide doesn't mean anything to anybody because that's insensitive. We're looking at plants which can sense that. And we're still thinking about the like ways to the particular thing we want to look at. Like one thing you guys were just talking about gas sensors, because we were thinking about making sort of a cheaper version of some of these physiological measurement systems in plants. But gas sensors are kind of more expensive than the sort of educational lens we have in mind.
But you might be surprised. just in that list, focus on what you want to manage. Don't worry too much as yet about which technology will
Nice.
come in.
Okay.
Because that's the whole point of this.
Yeah.
Like somebody can say mass spec, let's do mass spec for $100. So, you just have to be open about that. Oh, I'm gonna focus on the physics of how that works. Remember Raman, the first Raman spectrometer was $2, right? So it's kind of this, it's an artificial idea. Like this gas sensor is a laser pointer and a detector, that's it. That they're building.
Yeah.
So just, you made me realize something, this is totally side comment, which is, you know, we believe plant physiology changes as a function of CO2. They choose how many stomata they will put on their leaves or not, how much water they're losing. So it's literally evolution at play. And it just made me worried, or thought, do human physiology also change? Like in our lungs, has something changed from 200 to 400 ppm? It's interesting because at least from a percentage point of view, it's not that, but I guess maybe because we don't depend on CO2 in that way.
I mean, it's just the
It's
quantity.
an interesting idea. Yeah, like the pH of the mucus of our lungs.
Well, I know that at higher elevation, they have higher hemoglobin content.
That's correct.
because-
Yeah, so but it's all oxygen we
Yeah.
react to very quickly. I was wondering as a human, it's just a fun idea. But I love the educational part of it. What if like if humans were plants, this is what you would start seeing, right? We started, what's that movie in Marvel? The guy who's very rocks and scales on his skin. We need a plant man for Marvel. I really need it. I'm not joking. We need a plant man
Like an
that reacts to the CO2 of the world, that he's angry because he has more stomata, like pimples coming on his skin, and you know, I think that's something there. Yeah. So I mean, I love the educational lens. So I think it's very important to kind of anchor that oh, what we are building is going in a high school. Like just a high school in Palo Alto, so you know who your audience is. Then if the experiment has to work. Now the other pivot to put or anchor to put is, oh, it needs to be something that has a real plant in it. It's not a video, it's not a YouTube. It's a anchor that you put. It's not about, not other ways are not viable, but just that we anchor in real plant physiology, because you need to, again, remember, experience versus knowledge. And lots of YouTubers can do the other part, but you know, only some of you can do this part that, oh, we got in here with a plant and you showed the kids that plants breathe. Like, what if there was a pulse that we're not measuring? And the work to read here, first of all, just historically, there is a remarkable physicist called J.C. Bose. He's known, he was the first person to show that plants move. And then he built these, actually, let's show it. Can I connect with my room?
Yeah.
It's the most remarkable hidden history of science lesson. Can you all hear us, by the way, folks that are online? Thumbs up, yes, no? Yes. Okay, you guys, who online and folks connecting from India knows JC Bose? Where is the browser? Firefox. Do you want to turn this on? So I would say he was the first person to measure the pulse of the plants. Share screen. What's going on? Usually, oh, you did something. Green. Okay, I love that. That TV screen says quantum view. Is it quantum? What is not quantum? Sorry, we're joking about our TV screen.
Extension, so you might need to drag it to one side.
Okay. But I already did share screen. Share. Are you guys seeing something? They are seeing it, but oh. You are screen sharing. Okay.
Yeah.
You guys are seeing something? You are, okay.
Let me see if they if you drag it on.
Now you still see something? Ah. Can you all still see something? Frescograph? Yes. Rupali says yes. Oh no.
Okay. They changed it.
Just do the.
We did see it, and then you moved it away.
Got it. Got it.
I'll share this second. Okay, they should see this screen now.
Okay, you guys see the screen now. Frescograph. Okay, so J.C. Bose, Calcutta physicist, he decides and believes that plants move, and he was not the first one at that time. You know, Darwin had done a lot of work on that, but he was the first person to build instruments that could measure real-time physiology of plants. You know, it said listen to plants. So he built instruments that would measure sound in plants, and actually something that you all can do from a CO2 and then demo. It's not a good demo for a classroom, but if you live in cold areas, you take a stethoscope, like a normal stethoscope that you go for, because I'm reminded this because of listening, and you walk when the winter is just arriving, and you put the stethoscope on the trunk and you put it in your ear, and you start hearing these popping sounds. That's the capillaries in the plant that are breaking because they're above now the total surface tension pressure with that much cold. And every break is that vascular channel gone. And so the total flux coming in, it's almost like your core arteries are breaking because there's an air bubble now, and you hear this massive pop. And then when the summer arrives, the plant has to reconfigure its tissue inside. It's a really beautiful set of papers to rebuild those. So it's kind of like angiogenesis in plants to recover from an overwintering. So you can hear this, but I think kind of historically what's valuable and important is read through this, and some of the instruments that he built are really, I mean, incredible sets of instruments. And there's a couple of history books that I have on him. They have not really been recreated. Some of them are quite sensitive. And so there was always this notion of, did he actually measure? The instruments were so sensitive that he could play music to a plant and get some signal. Now, that's been very controversial. This was sort of some of the last work that he did. I mean, but this is 50 years of work. It's a long time. And kind of this, no, I mean, I can imagine mechanotransduction, vibrations. Yes, you can hear certain sets of things. Whether you can interpret that data, what that means. So I think kind of reviving it back, and kind of bringing him alive, and kind of also a little bit on the individual as well. When you're thinking about anchor an education project in a historic perspective, he would be a phenomenal character. I mean, he could be the Marvel plant man if we want to think about, and he shows up at a classroom, and then one of the threads would be is go through the review of these instruments. It's not that, I mean, it would be really fun if somebody wants to. I would love for us to build actually one of the and of course, this is not the only instrument he built. He built many different things. But Life Movements in Plants is kind of the key book to start with. Separately, there has also been, I'm just going to unshare my screen now. Okay, can you guys hear us back? By the way, all the discussions about projects we're having, if people want to chime in and contribute ideas, you should just raise your hands. Going back to stomata, there is an old project that we've been doing, and I've been wanting to involve Dominique and a couple of plant biologists on campus for a while on this, is a full scope based community mapping of stomatal density.
Yeah.
Super simple. You take nail polish, you walk to any plant, you paint it, it dries, it makes a little thin peel, you peel that off, you image it in a full scope. Oh, we haven't given full scopes to everybody yet. Okay.
Some people sent addresses, but
yeah. Anybody who wants it, just post it online.
Yeah.
But then everybody should get that pro kit that we have in the lab. You know, the one with the pouch.
Yeah.
Yeah. I think we should just be playing at this point. Also, let's do a film screening for Observer. Did any of you see the film?
I'm gonna see it.
Okay, nobody was able to come. All right, so I want to do a film screening. I think it's important. Let's do that as soon as possible. We can just do it any evening. Would evening be a good time? Yeah, and I think it would just be an important philosophical thread. It'll also anchor a lot of educational projects immediately. And then we can have students online also. Maybe we do that this Thursday. Okay, so the comment I was gonna make was this is really around citizens measuring density of stomata over time. Kind of long time ago, a Microcosmos post that I wrote, full scopes couple to leaves with your cell phone then, and you can leave them over time. So I have these data sets where I can see the sun is rising and the stomata is opening in real time on a live plant outside. Like, so you can start thinking about that. Oh, now that was imaging based, and that can also be done. It's pretty straightforward. But I am thinking of either using speckling, because speckling is very sensitive. Remember the optical cryptography thing I was talking about? Imagine a speckle pattern of a broad beam on one side. So speckle is just things where light matter interact. You get all kinds of these scattering, and you're imaging the scatter. And can you correlate that scatter change to actually force opening and fluxes? And so my intuition is that a speckle pattern of a leaf will encode certain aspects of physiology. It's a super simple setup. $1 laser pointer, $5 webcam, and you're not imaging, you're just catching the speckle pattern. And of course, it'll be sensitive to things that are moving, so no movement. So we anchor it to a point that there's no movement. But then we watch, you start with a very dry plant, and then you give it water, or you put it in a high CO2 environment and kind of see. And just starting to think a little bit about other clever ways of monitoring. Microscopy is totally one that would be awesome, but other clever ways of monitoring stomata opening and closing.
Yeah.
Because it's in some sense, even though it's not real time, I've always puzzled with what is the fastest time it could, like if you give plant a CO2 shock, you put it in a chamber and you turn the gas on. Would it is it 5 minutes?
Seconds.
Huh?
I mean,
Seconds.
the actual photosynthetic response.
Yeah.
Yes.
Yeah.
It's
No.
just the amount of time.
But the CO2, the stomata, stomata cell closing, because that's like the most mechanical large scale movement I see. The other part we could see is we could see the photosynthetic response. That would be another technique that we use in diatoms actually, and it would work in plants. For diatom health out in the ocean, we use a spectra where we will excite at a given wavelength. And this is also for symbiont health.
Ham.
Yes. Ham would give us the decay length and the decay time scale for a response. And it's classic in diatom literature. And
For leaves, there's always the FBFM.
Yes.
Yeah,
So-
yeah, Pam used that,
Yeah.
sensor.
Yeah, but what I'm saying is building a FEFM sensor would be trivial.
Okay.
Yeah,
That's one thing I.
FEFM, we could just build, and I think
Yeah.
for planktoscope, we are right now putting it inside a planktoscope because we have all these plankton coming by, and you want to not just know it's alive or dead, we want to know its health.
Yeah.
So 3 different things, speckle patterns, full scoping on a leaf, and then the third one would be FEFM for a real time. And then you have a place, you put it in a glass jar. It's also a little bit about with instruments. If we go to the JC Bose route, things should look a little old and from somebody from 1900 has come about. So that's where the bell jar comes. Like education has to have a little bit of theater. This is important to remember.
I think
Like you have kids, 10-year-olds who have absolutely no interest in what you're gonna say.
Megan had a
Yeah.
great point, and I like some of this stuff. Like
Yes.
listening to these things is if this thing just spits out a number.
Yes,
that's not
nobody would
useful.
care.
It
Yeah,
needs to be.
yeah. So
sensitive.
think of it as a art performance. Your
Mhm.
output is kind of a experiential art that you're walking out of.
Mm.
And it's a little bit about, you walk to a doctor, kind of feel, oh, I'm getting a pulse. Okay, so awesome. Build a team around. I think there's plenty directions to go and kind of. So in the Fred Park, have these 3 or 4 technologies. What's super nice about them that you don't have to invent any of them. They kind of already work. And then there is a layer to it, which can be tweaked. And then there's lots of optics people in the group itself. And we are anyway building a FEFM sensor for planktoscope. So we've just done a Raman probe. It kind of works. We still have more work to do, but this current instrument that Hannah is putting together, and she's not building the optics part, but I have to figure out. I'm gonna recruit a little team, maybe Enrico. But the thread around this would be is the same instrument probe can actually be used.
Mhm.
Okay, so I think this has all the hallmarks of the key sets of things.
Yeah.
The idea would also be is try to choose a species early.
Mhm.
Like this is the plant species. So you standardize it early and then you say, oh, it doesn't matter. Yes, Danny.
On the human physiology point,
Yeah.
I've been thinking, I think we actually use CO2 concentration in our blood, or like in our blood,
Mhm.
then to know to take a breath, not oxygen.
Mhm.
So I bet there actually probably is a difference.
Yeah.
versus
It would actually be fun to figure that out. It's a nice thing to say explicitly about climate change, that it's not just climate, you have changed.
Mhm.
Yeah, no, I mean, CO2 is in our blood. I mean, I'm thinking pH.
Yeah.
A common thing that might come as surprise, not to Sean, but to many other people listening. You all have oceans running in your blood. What I mean by that is the potassium and many of the key salt concentrations are exactly the same as seawater. And so it's not a surprise when the transition sets of happen from a physiological point of view that organisms and cells had evolved the kind of osmolarity and certain sets of things to be bathed in the ocean. When they started moving out, they maintained it. Evolutionarily, it would make a lot of sense. And so ironically, many of these sets, so it just sounds nice. You have oceans running through your veins. Can never go far away from them. Okay, project ideas. Yeah.
I guess there's kind of 2 I'm thinking about right now.
And then can you remind me your name
I'm
again?
Daniel.
Daniel, yes.
Yeah, so first, I'm actually pretty interested in the Planet Pulse project.
Yeah.
Over summer, I'm doing a like across America and teaching students.
Fantastic.
So if we could
How
like,
far are you gonna go?
we're gonna go to DC
From here?
and we'll
Okay.
do like I think.
I have told the Stanford group, and they have done it a couple of times.
Yeah.
We stack the bikes with foldscopes
Oh.
and drop foldscopes along the way. I think 3 or 4 groups have done this in the past, the Stanford groups. And then there was a biker who biked from Laos and Cambodia, and he wrote a book about it. So as an inspiration, I'll give you his book. And as a separate, think about that as a, okay, now you should tell me.
Yeah,
Yes.
so I guess like we're doing like I think 10 or 15 teaching sites along the way.
Mhm.
So I was thinking it'd be pretty cool if we can like deploy the project directly in summer.
That would be
See
super
how all the
awesome.
students react to it.
Yeah, and then do you guys already chosen the schools?
Yeah, we're in progress
Yeah.
choosing most of them. We have an idea.
So then you're already in a team then?
Oh yeah, team is established. Like I'm.
Yeah, no, I didn't mean the bike team. I mean,
Oh.
so you're joining the club?
Not
No.
for sure.
Oh, you don't know? Okay, this is one
I haven't,
possibility.
like-
Got it. Not convinced yet. You have to do your job. Yes.
I think.
No, it's very important to recruit
Yeah.
because the goal is if at the end of the class we have some awesome Fred Parks and ideas, we didn't do our job.
Yeah.
Like I want you guys to be at this point is like, holy shit, what am I making today? Like time is running out.
Yeah.
And it's very important to actually try stuff. because when you anchor and try stuff, it gives you the first hint for, oh, this is gonna work or not. Okay, so that's one, but there is an alternative.
Yeah, I think the other one I'm interested in is there's a on the idea board about measuring microplastics using a plectoscope and
Mhm.
trying to make like a super low cost way to do a measurement of some water sample that you're interested in.
Mhm, mhm.
I think it's definitely super useful either.
Yeah.
Even
Yeah.
like at your house, like oceans you're going into, your pool,
Yeah, and
water you're interacting.
I mean, what's super cool is microplastic is in much, even in our household water, much more common than we think.
Yeah.
I don't know if you guys read the tea paper. No?
It's
Tea
like,
bags.
millions of microplastics.
Exactly. Tea bags are made of plastic. It was a recent move. And people are like, oh, no problem.
Yeah.
You put them at high temperature, they start leaching out plastic, and there are literally billions of microplastics in your tea.
Yeah, I thought I was like, oh, good.
Yeah. And so, but imagine if you could measure certain things like that.
What were they previously made of?
Huh?
What were they previously made of?
I think cloth or paper. I don't know. Yeah, I think paper tea bags is such a common thing. And cellulose we can handle, plastic we cannot.
Yeah.
And I don't even know, like, what was the point? Why would you even move? It's not even more cost effective. I think they wanted to make these triangular ones so they hold shape. Okay, so that's super awesome. It's not totally crazy. The thing that I said about the FEFM being on a planktoscope in a flow-through system. For the flow-through system, it's not in a planktoscope incorporated yet, but we actually built a laser-based system some time ago for a planktoscope that is a trigger-based system. So it doesn't just image, it only image when an object comes by. There is a laser, and all we need is fluorescence. So just incorporating fluorescence in a planktoscope would be a super awesome project. It will naturally cover all the other things. It's a channel that is already needed. All the infrastructure is there, and we hack together. We have 8 planktoscopes sitting in the lab on a shelf. I can give you one of those. Build a team around it so you're not alone.
Yeah.
And microplastic would be one. And what's very important with autofluorescence is you can correlate some of that to even type. And then there is a second portion of, which is, if you achieve that, then you can think of Raman. Because if you can do Raman, you can actually tell exactly what plastic it is. And then one thread is we tested this, the Raman idea, this strip on microplastic at sea. So we have our tantalizing first data, which is useful. There is a third thing, and I don't know how far that project had gone. It was from MIT at Hui, a student group did measuring inflow impedance response of plastic. So impedance response, just think of capacitance. So there was no imaging chamber, nothing. It was just 2 copper plates and water was passing through. But then they had a fairly sensitive, it's called the 2-point probe, which sends a RF pulse and receives an RF pulse, but you're effectively just measuring capacitance. And if the probe is small enough, when you have a dielectric come in, so it's a dielectric response of plastic, and they were able to correlate for macroplastic, the dielectrics were compared to which material. Now, nobody's been able to shrink that down, and most plastic that's hard is micro, but for a lot of applications where there's macroplastic, that's super useful, like recycling, for example. You say, hey, what is this? I have a shred of this. What do you do with it? Most, many times labels, we say labels are put, they're all gone. So this is sort of separate, but it's useful for you guys to read the papers, see how far that project has gone, and then document it in a manner. So I think it's very important. Let's did we choose a date for the first project presentations, like mid-semester project presentations? How about Friday we do that? Would that, I think, yeah, it would force your hand. Yes. Okay, for people that are not there, let's just declare Friday morning we do our first round of project presentations. Everybody needs to be in a team. So I don't know if you guys all heard it. Friday morning, we're going to do final project teams, it's going to be the first when you are explaining what you're doing and race to the end this Friday. So the Friday morning session. Okay, I think, yeah, you have to choose, choose a team, kind of, I think either way would be great. And it's also, I think you can do both because you can let these guys do the work. You just take it on your bike for the plant side, which is great. You need that because you need to test. So that's called the validation phase of a project, where you see whether it was a total flop. And it's good for you because you won't be there if it didn't work. It's like, it's your fault. Okay, ideas.
I'm Peter.
Yes.
Uh,
Hold on, wait. Peter, Daniel, Danny, Jake. Jack, wow, so close. Jack, Enrico and Kevin, yes.
I have the TA on Friday, so I'm unable to make it to
Okay.
the live session. So
Yeah,
I'm pretty
yeah.
bummed, but
Yeah, but you catch on the videos, the video.
yeah, it was.
Okay, you have some
Yeah.
videos, yeah.
Okay, so I haven't done the Fred Park for this idea. I'm hoping to develop it more by
Yeah.
tomorrow.
No, I think it's good.
But I was thinking about waste conversion ideas, and I went to this. Obviously, I told you guys in my background working on fungal lab,
Yeah.
thinking about fungal waste conversion, but just generally trying to get at that problem. I think that we're working on the basic science technology development side, but I'd love to approach the problem for what it is. So agnostic of the method. But I was thinking, I went to this really cool kind of like art demonstration. There was a seminar that was kind of like a closed group application thing, mostly for the fungal community on campus. But an artist came in from Ojai, where she makes these really incredible weaves of human-made materials, lots of plastics, denims, things like that, and then grows mushrooms on it.
I was on that.
I have a picture. I was looking at her website. They're huge. Like she does these floor to ceiling, and then she incubates them. And so part of the workshop was she had a bunch of scrap material, and then we made things, and then
Let fungus grow on it.
yeah, it was so I was trying to think of ways to basically turn that into more of an accessible frugal education module. It runs into a lot of the similar problems of these things grow very slowly. And so there's very little. I mean, like I think it's a pretty stark image if you can bring an example of like a plastic mass with beautiful oyster mushrooms growing on it. Like that snapshot is cool. But
it's more of an end result.
Yeah, she tried to do this where she gave us mason jars that were sterilized with feed in them, and then we inoculated them with like a big syringe. So there's like things that probably would not go well in a classroom, and also would not like ship well. So I was trying to think of ways to modify that, basically have a little more tactile hands-on approach.
Yeah, so I think a couple of things. I mean, first of all, you're anchoring in education. That's good. It sort of starts. Choose a group, like an age group. And I'm thinking, you guys plan and pulse of a plan. If you go a slightly older age group, like,
Everything high school.
yeah, high school, and then you can think younger. I'm saying it's not that you have to, but if you divide that into two groups, like the comment you made about syringe, suddenly if it's too young a group, then that goes out. So choose an age group, and choose an age group first before choosing what you want to do. That's the key. Like, who do you want to inspire around this idea that human-made materials are also decomposable?
Yeah.
I mean, that by itself is a puzzling thing. And ironically, a lot of things that people say are biodegradable are actually not biodegradable. This has been a huge lie. So lots of people have done these soil studies to show that, oh, the plastic that was labeled as biodegradable is not. So it sets into this space of, hidden world of what happens to your waste. I can imagine many different ways. And one way to bringing, again, life and action on the real time is, again, microscopy. Fungus is so dynamic at the small scale. And so both Foldscope, several other scopes that we have in the lab, fairly easy to set that, in this piece, you're seeing the slow stuff. And what if you flip it around and then the other hand is a live microscopy image of what's actually happening? Well, you didn't see it. You look at it as like nothing. It's totally boring, kind of like plants. You just flip the piece. So if you look from this other side, then you're seeing all of these nuclei running around. for the same piece, it's the same species. I mean, that contrast could be one way of elevating. And one suggestion is, I like the inspiration from the artist's work, but don't replicate it.
Yeah.
Like go in a totally new direction, because the purpose is that you're trying to evoke the same emotion, but in different ways. But you bring a scientific lens to that problem, and it's sort of, I mean, what she's done is shown that something like that is possible. But it's also useful to kind of reset, so you're not bound to fixing the problems that might exist in that work. And she might have made other decisions around the artistic value. You know, like I was in that talk and it was about smell. It was a funny thing because this now people wanted to show it in the art gallery. And then they're like, no, take your piece away. It smells too much. It's like, what? You wanted it in the art gallery. It's a live mushroom. It's going on plastic. It's so beautiful, but only in photograph. When you come close to it, it sticks. But that was the wrong perspective. Maybe it should not be in an art gallery. It should be, you know, you should change the perception around what does it mean it sticks. You should actually do something with it. So people end up putting bounds around what they do. And it's valuable to just strip those bounds away. And for all of the projects, you know, use people as inspiration. So when I say Bose is your inspiration, but you're not grounded. You have to do something new. You have to do Bose if he was here today. Not, and I mean, it's an interesting side thing to recreate history for other purposes. And that's why we're using lasers. He didn't have lasers. So, I mean, he built purely mechanically. He built the types of things and subtle cell growth. I mean, when you think about it, cell growth, I can give a number to it. And why is it? There's no fundamental limit to rate measurements mechanically. But if you had many other means now with lasers, you could do far more. So, like, Bose with lasers. We have there is a comic there. Okay, so I love the idea. And then the other part of this would be is, in some sense, choose a particular substrate to begin with. Like in that artist will, like, she went from denim to this, but this was like 20 years of her work. So I would say, don't choose the mushroom species or the fungus species, but do choose a particular object that evokes emotions in people. Like, what is this that people don't like throwing, or what we throw? Like pencil is a funny example. Pencil has wood, it has graphite, and like, now if I had termites growing in pencil, it would evoke a kind of an emotion that kids understand.
Yeah.
Like their tiffin box, something that they often would use, but just choose one object and like only focus on that object. It'll get the mechanics going. Yeah, I think backpacks has to really be like, always the kids ask, hey, Dad, can we buy a new backpack? Not because there's a hole in your backpack, but just because now there's some new cartoon character that has come about. And so kind of viscerally showing what happens to your backpack when it's gone. Yes.
What about like shoes?
I love shoes as an idea too. I think shoes goes in all generations. I was thinking something that is so symbolically school, because you want teachers to take that on. And so yeah, I think it would be phenomenal. But again, shoes is the same story. Like we do have this scenario of like, oh, I think shoes, I do know for kids, they grow out of it very quickly. Backpacks, they don't, because they grow so quickly. Then it's like, yeah, it hurts. It's time for a new shoe. Okay, so phenomenal idea. I think you have kind of the pieces, build a team around it, right of Fred Park. Don't do it alone. I think it's kind of useful, and try recruiting some of the people. Choose a context. I mean, you can choose two contexts. So if somebody from online joins the team, so you can choose an American middle school and somebody else chooses another. And so there might be two objects then. Yeah. Okay, anybody else wants to mention projects and progress they've made on their projects? Any team that's solidly growing already? No? Okay, remember Friday, it won't be an option. everybody will be presenting teams and what you're doing. So this would be the time. If you want to put some things together, let's try to put them together now. Okay, let's transition to what I wanted to talk. Oh, Indumita, go ahead.
Yeah, I haven't started the analysis, but I've had like a few people ask interest, but I'm not sure like where to start.
Yeah,
Mine was
this is the
the
discussion we're having. We're trying to kind of narrow down, and this is a brainstorming session. So share what you are thinking about, and everybody will build upon it.
So I think the main thing is.
And then can you introduce yourself just for context?
Yeah. So my name's Indumita. I'm a master's student at Imperial College London, studying bioengineering. And my project idea was using ultrasound transducers, using the pressure waves generated from them to actually create a virtual lens inside the tissue.
Mhm.
So you can optically image maybe like a few millimeters, centimeters, I'm not sure yet, into the skin. And last time you suggested applying this to the disease called river blindness, onchocerciasis.
Yes, onchocerciasis.
Yeah, that one. And so I'm just trying to figure out the logistics of, they've proven it works, but I don't think they've tested it in like on like humans, like, so I'm just trying to figure out like how it would work.
So the way that I would say and structure the team is, let's actually anchor the team around a question. And we have worked on this. We have clinical partners for this. I'll send the team some pictures from my last visit in Cameroon. You walk into a clinic, it's a surgery clinic, and you have 10 kids sitting outside the bench. They have a little mass on their hand. It's a little bump. And the doctors, remember, hands are a very tricky place because hands have nerves. So you might say, oh, there's a mass growing at this knuckle, might as well remove it. But if you cut the nerve there, then the finger is gone and hand movements are gone. So it's very tricky. You're making this decision. It might just be a bump. It might be a dead onco, and you don't need to remove it. But if it's live, it must be removed because otherwise you get in trouble. So that's the challenge. Then suddenly you have anchored around the problem. And initially we're not starting, and it could be growing on your head, it could be growing on your neck. Let's just start with hands. That gives you all that you need in terms of depth, and then I would list all possible technology. So I love what you're describing on the ultrasound. Share that technically, how that works. But I would keep the tech space a little bit open. Maybe there's a very simple probe for elasticity. That elasticity in a particular way could give you. Maybe we can measure the pulses inside this worm. So just for context of the problem. Now, if you are squeamish, look away. But this is the reality. So it's if you want to see the so I'm gonna share screen, desktop too. Right. Okay. People can see my screen. Okay. Onco. So. Okay, onchocerciasis nodules. So the worst case is when it shows up in your eye, but you want to catch it at this point. You want to catch it, and again, these are very, very clear, but you have this nodule, a person shows up and you don't know what it is. But in the context of onchocerciasis, worm cross-section tissue. So literally that little bump that you saw, can you guys see the screen still? Was the histological section, and there is the worm. This is the cross-section of that worm. And now you see multiple because it's kind of folded in and around. A massive organism is literally living right in there. And then of course, it's the one that's producing many baby parasites that are going in people's eyes. And they're blind because you can literally see parasites going through your eye. So it's a pretty scary disease. It's still very common. And one of the important things around surgery of what we are trying to do. And so you can see, people get some sense of thinking about ultrasound. We started doing the capacitive imaging, just the idea that I told you about microplastic. So next time when we meet, I'll share the deck on kind of the work that we started. And I think it's not that you have to choose this disease. That's not a requirement. But my suggestion would be is choose a disease, because it'll give you all of the anchor points that are needed, and then scan the sets of technologies. And then, remember, we say often image the tissue, but image the tissue for what? What's nice about this is you are anchoring it in diagnosis. All I want to know if this is truly onco, and I want to know whether this worm is alive or dead. Because if it's dead, it actually decays on its own, and you might not have to operate on the person. So the criteria is that, do I operate or I don't operate? And so then it's not about resolution, or it is about some resolution, but not about a gorgeous picture that a scientist would care about. Because that's a broken, like ultrasound, it's not about creating pretty pictures. It's about understanding what's happening to a pregnant mother. So I think it'll change your feeling about even whether it's what are you trying to do an image. And it might turn out that an incredible resolution image is exactly what we need. And you don't have to choose onco itself. You could potentially also choose others. It's just that I'm saying we have an onco network. We've been working on this problem. I didn't have the bandwidth when I did the first trip to continue. So it's just still piled up. I still have to publish the original data. And what's super awesome is we actually established a cattle model for onco. So if you go to cattle farms in Cameroon, you can buy the hide for free because they're being cut and sold for food. And there is onco, a similar species that actually occurs in cattle. So all the data that we collected, we did that both for humans, but we also made the same measurements in cattle. And that was cattle has too much fur on the other side, but when the hide is removed from the other side, we got the access, and it's very much like skin. So we demonstrated at that time that it's actually very similar histologically. So what's very nice about this is that there are very straightforward, simple ways of testing because there is an animal model system. Yeah.
Does it impact cattle vision in the same way?
It does. I don't know what the late stages are. I think it might also be how the immune system handles the total body size. And I mean, some of those river blindness occurs over your lifetime. So what you were watching are people like really, they've had this worm for 30, 40 years, just everything is deteriorating, but it comes slowly. And my intuition is that the cattle has a similar, but the vision is not the only place that's impacting. Vision is something that we are so sensitive to that literally parasites in your eye make you blind physically, but it's happening across the body. And I don't know whether they target the eye tissue directly or is there something, but this must be known.
Is it permanent blindness, or what if you get rid of the parasites?
Recovery is driven by, I think in some sense, you have to think about lens. It just depends on how bad the damage is. There are times where somebody will just see a parasite swim by their eye. So it's in early stages. And there are others, and then they come and say, it was like, I'm seeing something in my eye, but it's not totally deteriorated. It's transient because they're traveling through the blood circulation. And then I think from a drug point of view, a lot has been done, but what doesn't exist is for people who have this giant nodule. And many of these surgeries are also done, by the way, without anesthesia. So it's pretty brutal to watch. And I mean, if people want, I can share the slide deck of what we did in Cameroon. And it just, that's when it feels like, oh, you do this the person goes through this, they lose function in their hand. Oh, and it wasn't even an onco. So there is a cost benefit analysis around surgery, which is critical from a diagnostics point of view.
What else could the bumps be?
All kinds of things, calluses, people are working hard, injuries, there could be a dead onco. There are so many dermal diseases, and I think it's in some sense, the thread is there's no particular, like, even if when I was showing the photos, you saw some bumps are really big in a soft tissue, some are really scaly. It's not like I could take a photo of it and say, Mpox makes all kinds of bumps, Tungiasis makes all kinds of funny bumps. Maybe I should do a tropical disease lecture. If would be fun. It'll give the breadth of just what the hell is out there in terms of parasites. I think maybe we have to do a diagnostics lecture, but before diagnostics, let's just do parasites, because it'll give you the framework for all the other really. Let's do a Halloween kind of style parasites. Yeah. Halloween is not now, right? Okay.
It's a little bit.
Ways. We could do our own Halloween. Okay, that was fantastic, Indu. But have you formed a team? Is there anybody else interested in onco or imaging and deep tissue imaging in general?
There was one person who messaged me.
Oh, go ahead.
But I'm not sure what their real name is because I just saw them on Discord, so I don't know.
Got it. Yeah, please use your real name, people. Okay, that's good. And then I will, if you guys become active on Discord and tag me, I'll go in and put the resources and things that I'm talking about, so that way I don't become a rate limiting step. And then anybody who wants to talk to me one-on-one for projects, please let me know, because soon enough, the teaching will kind of divulge primarily into sitting and doing. So soon enough, I'm going to stop doing lecture light mode, but primarily just focus on sitting with teams and just moving things forward. But if you want that time, it's valuable to request it and do it early when it's useful, rather than when you've already kind of either you're stalled or you've taken a path. So I think this is in general, I know this is a makeup class and very few people are here, but just do let people know. Okay. With that in mind, how much more time do people have? We have another 40 minutes or so. Yeah, you're okay with that? All right. I'm going to use my computer, Kevin, so it's okay. Let's take a 2-minute bathroom break. And then post that on Discord.
And that laser gets back and
And it's actually very useful for symbionts too.
Oh yeah, that's why we would say build one. We'll use it.
We will. Inbox is not. Oh my. Yeah. Yes.
Manu, I see you're speaking at the SynBio for Sustainability Symposium tomorrow. Yeah, not until like 4 p.m. So, and I think you're just on a panel. I don't think you're. I'm really stoked for it though. This is been looking forward to this for a long time.
Yeah.
Oh yeah, drop the hammer. A full day of optimism.
I love all of the enthusiasm in the SynBio community, and I'm the realist that often walks in and kind of deflates all the excitement, but I do want to.
Wait, what? You're giving us in bio presentation tomorrow?
Yeah.
There's the
fun.
big symposium
Yeah,
tomorrow.
it's actually
From noon until 6. Yeah, I'll put it in the Discord.
Yeah, put it on Discord.
When you presenting?
He's on a panel at
I think there's like some discussion in the area.
4:40.
I was looking at these panels and stuff like
Mike Jewett or any,
4 or 5
Jewett's given the
minutes of
closing remarks.
something, and you feel like
Jewett,
not even enough time to breathe
Drew
or
is-
sigh.
Yeah.
Actually say in 4 or 5 minutes.
Drew is giving
Okay.
something. He's moderating some panel, I think. Yeah. A panel on how can synthetic biologists help sustainability. Who does? Come tomorrow to find out.
Okay, all right, I'm excited. Let's do this. Okay, how do I join in? Zoom is in Kevin's email.
I watched Chasing Coral last week too.
Oh, nice.
That was a harrowing experience.
How is that?
Yeah, exactly.
Is that what the toilet paper salesman? There's like the
It has the
Chasing Coral.
film, like
Isn't that-
filmmaker.
Yeah.
He was toilet paper.
Oh, the marketing guy.
The marketing
I
guy
don't.
was
I
a
didn't.
toilet paper
I didn't.
salesman,
I don't know what
and
he
wanted
marketed
to go
before.
And then he went to turn.
I to market toilet paper.
Yeah.
Get itself. Oh, Kevin, where's the link?
Yeah.
Because I think you did a special link for today, right?
Yeah.
What's up with the neon change when the coral becomes fluorescent?
That's the Simions.
I think it's always going. I think it's always.
Maybe fluorescent's the wrong word. It becomes extremely bright to the naked eye, and then it's like in a phase of death. And they were it's a whole section at the end there, and then they mentioned offhand that
that would be crazy.
they mentioned offhand that it was happening like a huge scale on the Great Barrier Reef, and then they had documented anyway. They were mentioning that it was offhand that it was something about like they were producing their own chemical sunscreen or something,
Yeah.
and then it kind of like moved on from that.
Bioluminescent bacteria that's actually taking hold. And that. Yeah, I couldn't imagine that it's only when you bleach.
Yeah, it looks like
Yeah.
it doesn't happen all the time,
Yeah.
but it was weird.
Recording in progress.
They didn't go into details. As if it was like some kind of adaptation,
Oh, interesting.
but
how
they still died, so.
I like my favorite part of that is when they've got Terry Hughes and he's like really upset about the corals and he's showing the salesman a difference between a bleached coral and a healthy coral. And the salesman was a really great intuition for like understanding people's kind of needs and understanding of things. He is explaining how bleached coral is still like white and looks amazing, but he wanted to like actually. He's like, you like the marine biologist, like, look at how horrible this is. He's like, well, both of them look amazing. And so he then needs to actually like, no, you need to actually understand it's the equivalent of
That,
forests turning white and dying.
yeah.
Bleach corals they still do look beautiful.
And dead forests can also look beautiful.
Exactly, that's why it's
Like
an
burned
interesting- I
landscapes
found that
can
a really
look beautiful,
fascinating way
yeah,
of-
yeah.
of needed to look at
Yeah.
when you come in and-
Yeah.
Like that still looks
Like, that's
amazing.
sick.
Yeah.
That's a problem.
Can I see? Oh, it's a photo of
He's
the-
got
let's turn it
big,
around.
big bleach things.
Yeah.
That's still,
Yeah.
if you don't understand the biology behind it, you
Yeah.
know, it's amazing.
Okay, so we're gonna start. I wanna talk briefly about compliant mechanisms and some of the mechanics threads. Share screen. Okay, so can you guys hear me? Thumbs up. Yes, you guys can see this. So this is, I think for all of your projects, you will often need physical substrates, structures, you need ways of anchoring things. Often for many projects, that is the whole point. And sometimes it's like, oh, I have a laser and I have a leaf and I have a detector. If you don't anchor all of that as a system, you got a floppy little thing that's just dangling around. There's no signal. For a microscope, you might say, oh, I have all the optics, I have all the electronics. If you didn't build the body, you didn't build the kind of structure. And then, so talk a little bit about a very clever, low-cost way of building mechanisms, which has been underlooked. It's everywhere around us, only if I tell you it's there. So I'm going to use an example. This may or may not have it, but it might. There's a lot of moving parts in here to begin with. So just opened up a squirt bottle for ethanol. It kind of stinks, and I'm starting to open it. First of all, there's tons of parts. I'm sure they don't want me breaking this, but okay, we won't use, it's still squirting. Let me see if this bottle might have, so lots of moving things that had to be built for a low cost. I'll pass this around. And you might say, oh, how do I think about manufacturing these sets of things? Similarly, let's use another one, another squirting mechanism. Lots of things had to be built. Again, has a moving part, right? Because somehow this thing, oh, this is the degree of freedom, right? Right? Like a dead thing is still pumping. Now, again, lots of moving part. And see, if I was to ask you, how many parts do you think, if you totally disassembled it, how many parts are inside and how many parts are in there? Because if you're manufacturing something, it's going to matter how many total parts that you build it with. So it's actually, I was looking for something and I found a counter example, huh?
I think 4 or 5, 4, no, 4 or 5 and a half.
So I know this is there.
One.
I don't think anybody's gonna complain if we open this, do we? It's funny, they make it to not be disassembled. Okay, I see a tube, I see another thing, I see a spring, I see this is another degree of freedom right there. There's another tube right here. And so and then I see a type of a, so I'll let you guys see this. Point that
There's
way.
like two chambers. Right?
Exactly.
Yeah.
There's 2 chambers inside. So if you were to cut this open, you would find tons and tons of parts. And why am I talking about total number of parts? Oh, when you wash your hands, track mouse stops working. Why would that be? It's my capacitance has changed. What has happened? Does anybody know why when you use Purell on your hand, your trackpad stops working?
Your hand was
Your hand's cold, or it thinks your hand's cold with the ethanol evaporating off of it.
Yeah, that could be. Temperature is not what they measure. This is really funny. What had just happened? Oh no, it's working. Okay. Software bug, not physical bug. Okay, so any comments on if I was to ask you, how do we do all people?
You do multi-speaker or
Sorry.
or gallery.
So the puzzle or a question is, oh, I want to do
Excel view.
no.
Show not video.
Yes. Okay, quick puzzle / question. If I was to say you have to build a squirt bottle for a sanitizer, can you guys see me? Oh, I can still open I don't have to hide. Okay. You have a squirt bottle, you need, what is this part is missing. But the extra challenge is build the entire thing as a single part. It's moving, but it's structured as a single part. So how would you do it?
Wait, including
This is separate. I want to mount something on top of this that performs the same function, but I want to reduce the manufacturing cost. I want to reduce, because every time you make things, you assemble stuff, it costs, when you do bill of materials, you would need these pieces. Every one of them would be roughly between, if you want to do injection molding, between $20,000 to $30,000. This little piece, $30,000. And of course, the total cost is not 30 because then the steel will last. This is tempered steel mold. So this will last for, I would say, a run for 30 to 40,000, sometimes 100,000 pieces. And if you're making house, if you have the capabilities, but if you're a designer, suddenly you don't have that set of a capability. So pieces start adding up. And that's why during the stage of production, you realize that, oh, I now have to worry about my assembly. And then somebody has to assemble all of this together. But so any ideas, how would you make these as a single part?
Ariane has a good idea. Just
Yes.
put a small hole in the cap.
Yes, but that's the ultimate frugal solution. That is a correct answer, one. But I was going for some moving parts. Still, okay, let me change. Build a one-part pump. They're both pumps, but I want to build a single part pump. So that could be our assignment for this week. You don't have to physically it. Can you imagine something that pumps, but that is only made of a single part? And now here's the trick. When I say it's made of a single part, that doesn't mean that I'm saying non-moving. And that's the point of what I want to talk about today is compliant mechanisms, where the idea is that we have a framework in how to think that that can move while still be single pieces. And so this is an entire design phase. And I think I just want to finish one thing I had forgotten to say a little bit in the Fred Park class is this plot that's also in Alex's book. What I like about this is, again, he is really talking about what I talk about, which is cost versus performance. That's the whole point of the class. And you can see he's done this for motors, and you could see that, you know, as you ask for performance, all curves kind of shoot up. And that makes sense to a certain degree. It's not as if there is a physical law. You can stretch this forever. But if you keep asking for more, you have to think about cost. But then you can see what happens in the world, that lead screws, ball screws, linear motors, they cross over in very interesting ways. And then there is that brilliant idea you come up with. That's why I was saying, don't associate what was there as a perspective of what performance you get. So all of you are in that red curve. The only goal of the class and the only reason we're doing all of this is to be in that red curve. Like, this is not a systems class. We don't care about just putting lots of things together. We actually care about thinking much more from the basics. How would you go about creating that red line? So I wanted to say this. This is sort of the end goal from a Fred Park point of view. So let's talk about compliant mechanisms. The first, I had talked about this, but it's sort of worth showing. And then I'm going to do this again to see if I can. I don't like seeing myself. Even I can see. So this was the Lego. The only reason Lego works is actually because of its compliance. So what is the word compliance? Compliance means an idea that there is an inherent flexibility in all materials, all objects. Nothing is infinitely rigid for just very simple mathematical reasons. You know, the world is built of atoms. When you think about every interaction at scale has certain amount of bond energy, and you get bonds, they deform. And that gives rise to compliance. Now, crystalline materials, amorphous materials will have different compliance. You can think about that. It's like metal will have a certain kind of compliance, and then plastics will have a certain kind of compliance. Wood will have certain kind of compliance. That's really coming from the atomic structure. But from a designer point of view, the reason Lego worked is because they were build these compatible compliance that lead to the amount of friction. You want just the right friction to break the brick, but not too much that a kid is really, the kids still struggle. They say, hey, Dad, can you open this piece? And that's because they are erring towards like, it's too stiff. And that's why Duplo is for younger kids. It's motor skills in terms of thinking about parts. But you could have made the actual adhesion energy between two Lego blocks arises from this notion of compliance. So yes.
How much workshopping do they do to get it just right, as opposed to the first time, or like when it first done, oh, that actually works out pretty well? Or do they like purposely do it just to the pull it apart?
So the original patent, it's all in the shapes and materials. And from the very beginning, that's what kind of stood this company apart. They really focused on both mathematics, understanding exactly where the 2 point. The first moment was just, oh, it's interesting, modular toys. Then it started as a toy company. They started doing well. But when these patents came out, it was about the ultimate mechanism that snaps very quickly, doesn't break apart easily. And, I think one of the threads around, and all of those are trade secrets in some sense, this was all done pre-modeling era. It's not they had computer softwares and other, they were intuitively designing compliant mechanisms. And just the degree of compliance and constraint. I think this is also a question of contact friction. There is a lot going on. But once like finish of a plastic will totally change this. And so when you're doing injection molding, so then they had to reinvent everything. This company is the most incredible injection molding company because out of their injection molds comes incredible finish. And that finish is important aesthetically, but it's also important. So anything that you would change, a slight temperature change in the plastic, slight geometrical misfit, small amount of burrs. Burrs are these little tiny bumps that can remain when you're removing an injection molding, would change it. And remarkable that for billions and billions of parts, they remain on that. So I think it's hard to kind of tease apart when technologies were built, but Lego as a patent is not a patent, it's a company of trade secrets. Infinite number of trade secrets, and that's why nobody can take them. Unfortunately, after all of that era, then it became a company of marketing. Then it all became about when the next Disney movie is coming out, which is unfortunate. Yes,
This
Kevin.
came around like when plastic was on the rise, they wouldn't have been able to do it.
That's correct. That's correct. And it was the material, the repeatability of the material. So this is not, I wouldn't call it, it's a little bit of a hint of a compliant mechanism. It's about compliance, that they are using the elasticity of an object to an advantage, to their advantage of function. That's what I mean by that word. And again, this is why errors cancel out and you can stack these and then over time you can build stuff. This was a paper that had come out, and to just show that what does the kind of manufacturing talk like, and then they actually showed by capacitive measurement, if you put them back over and over and over again, what does the repeatability and other factors look like? Okay, so
I think I read that
yes.
their design is so structurally sound that you can stack 250,000 Legos on top of just one until it collapses under that.
That's insane. Yeah, that's also something about the strength of the plastic using. Having said all that, Legos have taken over the ocean. There's a new book that just come out. It's called Lego Pieces I Find, and it's basically photographs of Lego blocks floating in the ocean. Like people used to collect glass. This is plastic. And of course, the book is about plastic pollution. Because it's become a symbol, it's easier to use to say all of this is also going in many other places. There are many ways of this thread, and it's a little bit now this sort of puts it in a perspective of contacts. And I think this is useful if any of you were building precision parts that you want two parts to click together every time perfectly. Like, so if now I'm just going to choose the example of a laser, you want a clip-on mechanism that whenever you put a leaf, the alignment of the laser is perfect alignment of the detector. But you want it to be simple and put that back together. You could start with Lego. But there is another entire world of what are called kinematic couplings, which make it clean in terms of the number of contacts. So what you're seeing there is if you want two parts to come together exactly the same way every single time, this is a little part you use. So you see on the bottom are these little bevels, and on the top is a hemisphere. Now, if you see when this hemisphere sits in this beveled groove, you get 2 points of contact. Now, if you get 2 at every point, you have 6 points of contact between the two pieces. There's 6 degrees of freedom. So now that you've just coupled all degrees of freedom of one object to the other object, that means that in this configuration, these two objects are now completely constrained. They're not over-constrained, they're not under-constrained. Lego is over-constrained because there are so many points of contact. So when you design an over-constrained system, you have to be very careful because any error will have clash between your constraint. So over-constrained can give you more friction. You can't just have 6 pieces because it'll just fall apart. So this thing, although it is constrained, if you flip it upside down, it's false. So it is not compatible with gravity. That would not work. So Lego is doing in that space of giving you just the right degree of constraints. But it's also, this is done in a cheap. And then there are other ways when you say, oh, I want 5 degrees of freedom and I want 1 free. And so then you're playing with this. And so, for example, the Kelvin coupling is essentially you have, now that is not a trough. You can see that these are little triangular. So then you have 3-point contact, 3-point contact, and an contact. And so for different functions, there is an entire class of these. And this is just to explain what Lego is actually doing. But they're doing this at a much higher level because you have so many points of contact. And then you do what is a field called tribology. You sort of measure points of contact. It actually turns out when an object touches another object as a sphere, it's not a single point of contact. It's actually a surface area because there is compliance. And that's why when you make these, so you reduce it. If I had made these feet out of rubber, all bets are off because then you have surface friction. Everything has changed.
Wait, how would it, with
Yes.
the cross, how would it be able to move much? Or I can't.
No,
Not at all.
no, not at all.
And also not at all with the.
That's correct. Both of them
Not
are
at all.
over-constrained systems because that one has 3 plus 3 plus 1. That has 7, and the other one has 6 exactly. And any object has 6 degrees of freedom in space. Translation, 3 rotation. You could imagine 2 troughs and 1 contact point. And then you would see and play around and you should be able to find 1 degree of freedom that would exist. And again, all of these are around objects that you want to take apart, put back, take apart, put back. The adhesion force can be magnetic. So a planktoscope, for example, has no kinematic coupling. Super easy to improve a planktoscope. We were out. Every time we use magnets, it snaps back in, but the flow chamber is not aligned to the thing. And then I asked Enrico, can you go touch it a little bit? I'm looking on the screen. He's shouting from there. It's like, yeah, oh, it just got it right. Don't touch anymore. Super simple. You attach a ball bearing and that thing, every time the snap force is coming from the magnet, but every single time you have 2 objects aligned. So I'm saying these things are everywhere and we just don't even think about it. And we don't, kind of my fault because it's super easy to do and we haven't done it yet. So now moving a little bit around, I think we talked a little bit about this. This was a context of, but remember in that, and again, going back to the voice coils that you guys had opened, remember the cantilevers?
Yeah.
Now that is an object that is a compliant mechanism. So a cantilever is the simplest example of a moving degree of freedom 1, 2, 3, depending on how you design it, but that's made out of single parts. And we'll dive in a little bit to see why we do it, what does that actually look like. And the oldest compliant mechanism I can remember that made a big difference was actually, I am thinking, I'm sure compliant mechanisms are even older than that, but this, if you guys know how gravity was measured, the gravitational constant, Cavendish, the famous Cavendish experiment.
It wasn't a giant pendulum, was it?
Yes. Okay, I gotta show it to you guys.
Oh, also, Scott's single cell masting is a cantilever.
That's correct. That's correct. All AFMs are cantilevers, single degree of freedom. Anybody remembers how we know the gravitational constant? A raise of hands. And what do cats have to do with it? Cats were very important. Okay. I don't remember. This is my vague memory. It might turn out that, let's see what Google thinks. So Cash did a really remarkable experiment where he would put, oh, let me just share my screen. He would put these giant balls. So this is the simplest example, and now you'll see why cats are important. Can anybody see a cat in this photograph? There is a cat, I'm telling you. Okay, there's a piece of a cat. It's not an actual cat, but there is a piece of a cat. Where do you see? Anybody online? Did you identify a piece of a cat in that photograph? Say that again?
Is it cat gut?
Cat gut, okay. You said something?
I did not say cat gut, but I thought just like the head and the tail. Is it spinning around?
No.
The sinew?
I really mean it, like there is a piece of a cat. You're close. Whiskers. Cat whiskers are an incredible torsional spring. So look at that photograph again one more time. You see this wire, and down here is this tiny short thin piece, and then there is a stiff piece. You have two objects of mass m, and you have two big objects. These don't move. The way you measure gravitational constant is you hang this thing, and you go and you change this mass to be larger, and you ask how much degree angle. So imagine doing this on Earth. Earth is super sensitive. If you breathe in that room, it's gonna bounce around for 5 days. Cavendish did this. He made a giant tower. You need a massive mass because larger the mass. But how much is the force? How do you measure it? And then the issue was, how do you make and find these sets of wires? So I'm actually surprised that the word cat is not mentioned here, and I'm hoping I'm not joking. Let's see. Cavendish torsional spring cat whiskers. Oh, come on.
It wasn't in a dream or something, right?
I could have dreamed it. I really believe that a cat whisker was the first torsional spring that was used. No, now these are cat toys. Let's look at this. So eventually he moved on to incredible wires. And the reason he was able to do this is because he was now the first person who could do, what is going on? These very precise manufactured wires. And the first sets of things that he used, I remember, was cat whiskers. I'm gonna go dig in and find. But that fiber that's attached is a compliant mechanism, because it's not using contact on how we think. It's using atomic rearrangement. So compliant mechanisms by definition have atomically smooth movements. What do I mean by that? Is that if I have a beam here, if you guys can see this beam, I bend it, I am getting atomically smooth. Right now, I don't have any control on it, but atoms are rearranging in this object to give me rise to this. And that's why by definition, if you have that kind of control, that's why LIGO uses all of these hanging wires and stuff, because they can atomically control almost to a certain extent. And you can write down first principles, where does elasticity of a beam come from, and calculate from bond energy, like literally bond energy. This is where what we call the constant of elasticity would come from. That given chemical covalent bond energy of two atoms, can you calculate first principles elasticity? And again, there are two regimes in this. I'm really stuck at wanting to know the cat part.
I'm looking for, I don't know, I'm trying to find it.
It's I love history.
His first torsional spring was a cat.
That's correct. Cavendish.
Did he own a cat or?
I keep trying to hit like those worms and all that seems stuff.
I really want to know. Oh, and the other clever thing that he did, which is also important, he used light in a very clever way. Because it moves so little, you don't even know that it moved. So you use an optical lever. So you shine a beam at it and you shine the spot, you mark the spot. The farther the spot are, the more sensitive your delta change would be. And so you can actually measure that. Okay.
They call that Abbe error. Abbe, the one who's like, oh, if you want to measure something, you should increase distance in decimals.
I didn't know. Yeah, you're right. It's
How accurate was his constant? How close did he get?
Oh, I think he was super close. He was super close. Yes. Okay, so I'm gonna jump to then I'll tell you guys about a problem per se, but now I'm realizing my videos are not here. Okay, now let's go to Disney's website and I want to show you me. Okay, so let me share my screen. So this is
Wix Studio, built to meet any brief. When your project calls for bold creativity, design freely on Figma and seamlessly move to Studio for your vision.
That's not what I said. Okay, I just want you guys.
We present a computational tool for designing compliant mechanisms that, instead of rigid articulation, obtain their function by virtue of elastic deformation.
Okay, so I haven't told you guys anything about compliant mechanism, but I'm just showing you the crazy world of compliant mechanisms first. Is that every rule that you know about design and how objects work, you can throw that out the window. What you're seeing here, and it's a super exciting time because this would actually be a super exciting AI interface. Because, you know, our imagination sometimes we're not mixing and matching certain things. So what you're looking at here is beams. Whenever you see stiff, its displacements don't matter. So you can ignore that. That's just a rigid object. But you notice the clever part that the two objects are connected to each other by these thin pieces. Now these thin pieces move. It's a very difficult mathematical equation. These two pieces, notice they're not even connected in parallel. There's a twist. So that object is both bending and twisting. If this thing bends, so I'm talking about this section right here. What a crazy thing to do that they didn't even connect. If they had just connected that way, they would get 1 degree of freedom. But if you twisted it, when this thing bends, it rotates. So there's actually torsionally the object balancing itself so that you don't flop in the other direction. And then if you twist it one way, the other one you twist the other way, you can actually get stiffness in the direction that it also becomes stiff in this axis. But this is an example of a crazy compliant mechanism that would take you years to solve and find, but you can just build it. It's a single 3D printed part, and it's a leg of a robot, for example. You're saying the motor And of course, this video is the design tools for these types of things. And this is a tip of the iceberg of what compliant mechanisms do. But let's just watch a few of them.
Our method takes as input a conventional mechanism and replaces rigidly articulated.
So what does that mean? Conventional mechanism, that's a pivot joint. So everything is rigid other than that rotation. So the way you would make this would be 1, 2, 3, 4, 5 pieces. You would connect the bearings in it. You have to put at every point, first of all, you have the certain sets of amplitudes, you have to assemble it. But rather than all of that, in a single shot, you get the same mechanism. And this particular video
flexures,
is
thin plates of
about
elastic material whose
I'm
shape
just gonna skip
motion
this
of an
too.
end effector tracking the target motion.
So-
However, this is not sufficient for
They're solving this computationally by saying,
However,
oh,
this
I'm
is
gonna
not sufficient
add
for
and
proper
slowly
functioning,
change.
as deformations in the flexures can induce large internal
Example of failure. So flexures do fail, is that if it goes above. And the tricky part is they both have linear and nonlinear responses. So to a certain And so the tricky thing is now there is also ways of actually using the nonlinear response of things, not to a failure point, but still nonlinear responses have all kinds. So I can encode memory in this, I can do crazy things just as long as I can design. And now the simulation tools have gotten so good. So we will, you can see here's a simpler example of a compliant mechanism. And the classic way that people have seen this as staplers were the first one to think about in terms of what the mechanisms have been made to be. So one thing that I want you to do is before we dive into I'll give you guys a key guide in our next lecture of compliant mechanisms course on, and we would still not complete it. So we're going to play a trick. I'm going to give you a sheet that just has design nuggets of just the key. So the simplest one is a thin beam. That's a module. A wire is another module if you want torsional springs. Now you can say there is a simple one. If I want rotation, but not translation, is a crosswire type of a beam. So there is a key card that's associated with all of the key designs people have built. Now, things like the James Webb Telescope, if you were to look, thousands of compliant mechanisms put in place because every origami is technically a compliant mechanism. Every fold is a compliant mechanism. And one of the key factors would be is to take the undisciplined approach of learning rather than taking an entire class. We'll just think of compliant mechanism as a zoo of animals or organisms, and then synthetic biology approach, combine those circuits together. You don't even know what you're making to see what comes about. No comment on synthetic biology, but just a comment on that is an undisciplined approach that we should play with. So we'll take that a little bit. And then for a subgroup that really wants to do both simulations and a little bit of a dive in, I can go and give you guys sort of one sub lecture on how to go from an idea to how to arrive at a compliant mechanism. The theory is a little bit involved. It is something that I was anyway going to go do for my group in some sense. So at some point of time, I would be excited about doing it. But what's valuable is now the theory is well evolved that we can actually arrive at mechanisms. So let me just, if I just type here, zoo of compliant mechanisms, blah, blah, blah. So suddenly, I just want you guys to just see and be in awe and wonder of just, holy crap, what's out there. So you can see all of these sets of things are crazy number of compliant mechanisms play. Now with AI, you can do things where you don't even know, you just optimize the network, that this object, if you pull it this way, it will go down this way. So you're converting one motion to the other, but you have no idea how you would come about it. While this is a very structured approach to it, this is what I was saying when I talked about rotation. You can see the cross beam here that it invokes the torsional. But every one of these things was invented by somebody in their head saying, oh, what would happen? This is a fun one. This is something that we built around. It's an XY stage. And actually, one thing I will show you is my class project when I was in grad school somehow is still online. Pocket milling machine. Let's see. When I search for it, I don't find it. When I search for the wrong word, I'll find it. Because I'm the only person in the world who spells Fred Park wrong. It's the first link. Because it's not how you spell it. So here is what I did long time ago. And I want to show you guys my project. What was I? Why was there I saw a Tintin there. I should document some of these things. Anyway, so we built this machine. The idea was, can you build minimum number of parts? I wanted to carry a CNC machine in my pocket. Don't ask me why. And now hidden inside is an XY stage. There is a Z stage. You can see the ellipse is the Z stage. And they all have different actuators. And then there is a mill that fits, which is essentially a very high-speed motor that just moves. And we actually built this. So this is
What's
the
CNC machine?
CNC is computer numerical control. So it can. So the idea is, and then you can see the coupling right there, the kinematic coupling between the two pieces. And you see here, for example, and you want to understand this. So if I push on this object here, you can see my screen. What do you think is going to happen? How do the stage move if I push on this? You can see there is a beam here. Now it's a double loop. Double loop beams actually reduce rotation, but just translation. That's why there are 3 beams. Yeah. Because if it was just one, I am rotating and translating. But when there are 2, you kind of bend straight like that. So you cancel translation. So that's where the double beam. So it's a module. Now, if I've attached it this way, I get this degree of freedom. But now I cancel. I also have this anchor here because I want translation along this axis. This mirror essentially cancels any other rotation that might happen to the stage. So you only get this axis. So the way that these things are nested are designed for making sure that I only get pure X and Y. And actually, there's a clever design that you can do pure rotation. So you can do 3 degrees of freedom just in this flat plate. And now going back to what did I, this is a single machine piece of steel. So you can take it and with one single shot, you've made all of these things. There are no bearings in it, which is super cool because then I have no friction. I have nothing that's moving against anything else. And so from a cost point of view, that becomes a huge savings. And again, as a field, it's matured significantly. And
Wait, so when you push down on it, it moves.
So that's correct. So if you move, where did we go? So this one, if you push here, you can see a hole here. Then I get an x-axis. And then if you push here, I get the y-axis. And then when you push here, I get the z-axis. Now there is a limitation to compliant mechanisms that sometimes distances are low. But I said I wanted to make a pocket milling machine. So I really wanted, this is designed to be the size of your sharpener, by the way. So like a pencil sharpener. That's the point of this. Sorry, it is not obvious. This is truly a pocket milling machine. So it's not some giant object. And so I really want to make very, very small parts. And if I'm going to make very small parts, why do I need a giant thing? And the purpose that I want very high precision, but not that much travel. Or there are ways of also doing long travel now. It's like that field has grown dramatically. But kind of the big thing that I want to tell you guys is the way we will do this compliant class is I will share that keynote, the zoo, but then we will, if somebody's project already it's obvious that you want to try this as an approach to design, we will just in real time designing a compliant mechanism for it. So this way, why do I? Okay, I think I have to put the key card together. It's probably, okay, I think time is up. One thread to do is read a little bit about compliant mechanisms yourself. And then Friday, everybody must be in a team. So, this would be the time to panic. Join a team. It's a contact sport. It's very important. You can play a very important role. We'll do a quick 2-minute open mic. If people have to leave, don't worry about it. But any questions? First of all, thank you for the flexibility. Just I had something that I had to deal with. But we will meet tomorrow.
Yeah.
We will meet tomorrow. Yes.
Tomorrow you have your Fed Park stuff.
Yes. By tomorrow, please do your works. We'll meet tomorrow. And then I might actually continue a little bit on, it actually depends. Give me feedback. If you guys really want to learn compliant mechanisms, we can really dive in.
We have a workshop on compliant mechanisms.
Why don't we do a workshop in compliant mechanism? That's a much more, I think I was starting to hope that all Fridays will now start to become project. And so let's see if one group can anchor their project becomes a necessity. Like we can try designing this optical levers. I want to do it in a context of a problem so that when we are brainstorming, we can do this in a context of some degree of freedom that's actually needed. It might be interesting, like a scan degree of freedom would be an interesting one.
Yeah, I just.
Let's just make a crestograph out of compliant mechanisms.
Deal.
Which whoever did. Okay, now I'm excited. Friday. Okay, bring some plans. I'll bring his book. I have that. Yes. Oh, I'm so happy what you said. Let's build a crestograph that anybody could laser cut. That would be awesome. Okay, we have our assignments. Open mic, any comments, questions? Are you guys confused about projects? Oh, go ahead, Shivam.
Professor, so this is something about like open problems which professor has shared. Am I audible? Hello?
Yes. Am I audible? Can you hear me? You can just call me Manu.
Yes. Yeah, sorry, Manu.
Yes. No. Yeah.
Yeah. So yeah. So I'll just. Is it okay? Like if I bring that problem, it's a bit mathematical in the sense it's about like. So it's just 1 minute intro. So it has got like different rates. It's about drug policy or making. So they take some genome sequencing and read, say, let's say, some representatives genomes or sequence, and then based on then do some sequencing. So it's about that. Maybe I'll ask professor, would it be possible to do that as a project? It's an open problem, but yeah.
Why don't you try making a Fred Park for it and then share that Fred Park with me. So anybody who wants comment on their Fred Parks, I'm happy to look at. All of us, you should open it, let other people comment on the Fred Parks.
Okay.
Like you don't even have to contribute to it. Just say, I read it. I feel I don't have actionable items as yet. Like, so then you would go back and say, oh, let me convert it into. So write it, share it. All problems are open. There is no, remember last couple of years ago, we tackled how to catch asteroids. So just do what we want. You want asteroids in cost-effective manner and clean up space, for example. So
Yeah.
problems, there's no bound. You want to make socks, shoes, whatever. It's but write a good Fred Park.
Sure, sure. Two more comments, Professor. So just, I think a friend of mine was telling that you have the project at Simla coming up. So I will be going to Himachal soon, maybe by June. So I was wondering if I can like be, if any way I can visit that place, the one which is, is there something which is coming up in Shimla by you?
Yeah, I mean, there are
correct?
full scope workshops happening around the world. If you're going to Shimla, send me a message. I'll connect you to the
Sure.
community. And I think same goes for you. If you want to just write to me
I'll send that
on the bike route, that would actually be super fun. Yeah, I am actually curious how many full scopes you can put on a bike and still bike.
Yeah.
That would scopes on a bike. That would be fun thing to say. I think we could do 1,000 full scopes on a bike.
Yeah, I don't know how big on it.
Oh, you haven't seen a full scope?
No.
Let me fix that
No, I
problem
haven't seen
right
it.
now. Sorry, I have to show this to you guys because you are all supposed to have them. And let's do that today, actually, at least, Kevin, for anybody who wants, just come by the lab to collect.
I would also like to have a full scope.
Yes, I'm gonna send those to everybody, basically. So I didn't realize that we have not done the full scope class yet.
I have to dash, but.
Yeah, go. It's all good.